Well, welcome everyone. Good afternoon. We're so excited to have you here in the offices of Innovation Partnerships to our very first event in a new series we're launching called Impacts of Innovation. First, I've been asked to share a few housekeeping notes. We are recording this session, so we ask that you make efforts after you finish your pizza, of course, to minimize noise, and also please save your questions at the end. We will have time for Q&A. To our guests on Zoom, welcome. Please keep your microphones muted until the Q&A session. And sorry you don't have pizza, maybe next time. So we created this series called Impacts of Innovation at a time of extraordinary momentum for the University of Michigan and for our role as a national leader in research commercialization. Today, Michigan ranks among the top universities in the country for innovation output. Last year, our faculty entrusted us with a record-breaking 673 new inventions, placing us second in the nation behind only MIT for inventions created. The same year, our team supported the launch of 31 startup companies, a category in which the university also ranks second in the nation. At the same time, our innovation pipeline is as strong as it has ever been, with 10 FDA-approved therapeutics currently available to patients and an additional 14 therapeutic assets in human clinical trials across over 100 sites. Our therapeutic commercialization output ranks among the best university in the country, making a difference in the lives of tens of millions of patients across the world. Our medical device pipeline is equally powerful, with dozens of devices currently available to patients today and another 10 in human clinical tests initiated by University of Michigan licensees and startups. This is another area where national benchmarking shows the University of Michigan ranking third in the nation. With this scale of impact and innovation, it can be difficult to pause and to fully appreciate what is happening at the university and to experience and celebrate the momentum we're seeing across our ecosystem. So this is why we created this event. Impacts of Innovation is designed to celebrate the real-world impact of breakthrough research discoveries and to create a space for us to step back and recognize the scale of what we're building together. This event is focused on giving us an opportunity to hear directly from the people who make the impact possible. The brilliant University of Michigan innovators who take an idea from the lab and carry it forward into something that changes the world. When my colleague Bryce Pills and I were envisioning this new series, we knew there was really only one person who could launch this event for the University of Michigan. I am therefore so excited that she said yes, and that I get to introduce Dr. Zhen Xu, the Li Ka-shing Endowed Professor of Biomedical Engineering at the University of Michigan and co-founder of University of Michigan startup HistoSonics. Zhen is the leader of a co-inventor of histotripsy and the leader of University of Michigan's histotripsy group, which you'll hear more about soon. What began as her graduate thesis project has grown into a transformative medical technology reaching patients around the world. I believe that histotripsy is on path not just to be the most impactful technology ever to come from the University of Michigan, but perhaps the most impactful technology ever to be transferred out of any university anywhere. Last summer, HistoSonics announced a landmark $2.25 billion management-led acquisition, placing them the second largest venture capital-backed medical device exit in history. At Innovation Partnerships, we've had the pleasure of supporting Zhen along her journey for many years, going back to her first invention report in 1997. In 2023, she and the histotripsy team received the University of Michigan's Distinguished University Innovator of the Year Award, which is the university's highest recognition for research translation and impact. Zhen also gives back to the community, and she is part of our inaugural cohort of Innovation Champions. a group of faculty whose leadership, creativity, and commitment to translating discoveries inspires others across the university to take that leap. And I'm so glad to see that we have several of our innovation champions here with us today. Thank you for coming out to this event. We're also honored to have other distinguished guests with us as well. Dan Braga is a University of Michigan alum and investor at Morningside with deep experience building and scaling companies across venture and private equity. We're also joined by Dr. Gerald Chan, co-founder of Morningside and a global leader in life sciences investing, whose work has helped bring transformative biomedical innovation to patients around the world. They both dedicated their careers to advancing the kind of innovations we're here to celebrate, and we were so glad that you happened to be on campus and were available to join us. Thank you. So with that, it's my pleasure to hand the microphone to someone whose work is changing not just how we think about treating tumors, but the very field of surgery itself. Zhen.
Thank you, Helen. Thank you for the invitation. Here is because I can't say no to Helen. All right. So today I will talk about my now it's like 25-year journey of inventing histotripsy and also HistoSonics. And also I just want to claim that people always ask me to share knowledge experience, and I have to say that I don't have much of that to share, but I have a lot of mistakes, lessons learned. So you'll hear a lot of good and bad, the true story of the journey. So just give you a warning. All right. This is not just a research talk. What I was told that I need to actually talk about this translation journey and about HistoSonics. But I don't have to disclaim the conflict of interest is about HistoSonics. To start with, I do want to briefly talk about histotripsy, just in case any of you haven't heard how it works and what it is. So the histotripsy when we first named it, so it was a term coined actually at Michigan. We follow the rule that all the medical terms actually based in Greek. So histo means soft tissue and tripsy meaning mechanical breakdown. So we are looking at a technology that can non-invasively perform the real surgery. What it means is actually mechanically destroy and remove tissue like surgery does, but without incision, without radiation, and without injury. So that's the idea. And how do we do this? So what we do is we actually I have to go back. I don't want to play that movie. Okay. So we use ultrasound energy delivered from outside the body, and supposedly when the movie play, it will actually show ultrasound generated micro bubble, and the bubbles actually is what kill the tissue. So this is a fast forward 20 years later. This is the first histotripsy clinical system. It's manufactured by HistoSonics and got the FDA approval in 2023. So what you're looking at here is that we have an ultrasound device that generates histotripsy. This sort of works. Okay, so there's an ultrasound transducer that generates histotripsy. And then in the middle, there's an ultrasound imaging probe and co-aligned with the therapy so that we can actually see the treatment. And then there's a robot arm connected with the ultrasound head so that we actually, before the treatment, we use that to scan our imaging across the tumor. Then the clinician, what they do is they outline the tumor on the ultrasound images they see. Then the treatment software will create a scan path so that it can automatically scan through and cover the tumor outline. And the treatment itself is automated. So after the clinician outline the tumor, then they've done their part. Then they click go, and the treatment will be delivered by itself. So what you're supposed to see is that there is going to be a tumor fragment in the gel. Then the treatment video will show that the focus will actually scan through the tumor automatically, and it's actually liquefy this. Then it's become this liquefied debris, and when we cut it out, it will just leak through. Okay, so you have seen the video now. Like I mentioned that you will have this tumor fragment in the gel, and then we have this pre-scan pass prescribed before the treatment. The focus is really just automatically scan through that, and then as you scan through, you can see that tumor is liquefied as you actually treat through. And after treatment, the operator will cut through it. And you will see this solid tumor, right? Now it's becoming this liquid, and when you cut through, it literally leak out. Okay, so in the body, and if you see this in person, you feel like it's a magic, and it is magic. And this debris that just absorbed by the body over time, and then you will be the tumor is gone, just like the surgery. So the benefits of this, so I'm going to, this is all kind of from human patients. Of course, now first is non-invasive. You can see that this is a tumor, right? And typically, if you do surgical resection, you're gonna have damage to the liver tissue. But instead, these patients receive the histotripsy, and 30 days later, that liquefied debris gets absorbed by the body already substantially, and there's no damage to the liver tissue. So really, the non-invasive. That without having to cut the patient open. Second, the high precision. So when you think about radiation or thermotherapy, there's always this gradual kind of not just damage or no damage. So you have high dose, high damage, low dose, low damage. With histotripsy, it's a threshold phenomenon. When the focal pressure is above the threshold, will generate bubbles. And you can see that within the treatment area, the cellular structures are disrupted. We literally are disrupted cells and cell wall and all the structures. And you can see that immediately outside the treatment region, there are individual cells. And here you have half of the cell that's remain. It's gonna die, but it's half of a cell and the other half of a cell actually getting cut off. The reason is now our scaffold is actually microbubbles. They are on the size order of cells. So we're getting very high precision surgery, and it's a threshold, meaning that above threshold, yes, below threshold, no. So really high precise. The only thing is we don't have the imaging modality to allow us to image the cell 10 centimeters down deep in the body to do that kind of high precision treatment. For non-invasive treatment, for an operator, the clinician's challenge is that they cannot see things happening with their eyes actually. But here with histotripsy, we are using ultrasound image as a real-time guidance. And those microbubbles are natural contrast agents in ultrasound. So we see them as a bright spot. So those bright spots behind the cross arrow, which is our focus, are microbubbles generated. And this circle is outlined by the clinician. So they are just watching during the treatment to make sure that bubbles are generated and scanning within their outlined region, not outside. So they still have real-time, visualization to ensure the treatment accuracy. Most patients after histotripsy treatment, they wake up and they ask, "Did you do anything?" They associate surgery with pain. Okay, so you wake up and you feel like this is no pain, and the doctor said, "Now you go home now right away, have dinner at home, and go back to your work tomorrow." There's nothing, like you have to follow certain rules. There's really nothing. So the people are not used to it. Okay. So the non-invasive surgery, because you're not causing injury, and you are still depending on body immune system to work afterwards. But with histotripsy, there is no to minimal pain and a fast recovery. Unlike a lot of the painful, toxic cancer treatment. And actually, the treatment by liquefying the tumor often relieve the pressure on nerves and reduce the pain. So the quality of life is actually really good. Tissue selective. So we actually later found out that histotripsy actually, because it generates mechanical damage. So the major blood vessels and nerves and the bile ducts, they are actually naturally more resistant to higher mechanical strain because those critical structures have a higher mechanical strength. Our body designed that way. Therefore, they are more resistant to histotripsy damage. And we are able to treat really engineer the structure or include the structure in the treatment area without actually damaging. So this has allowed histotripsy to be used to treat, for example, a tumor sandwiched between vessels without damaging the vessels. So, it's kind of coming from the physics. So I was one time saying that, looking at the imaging guidance, the bubble just naturally you can see them on imaging, looking at the tissue selective property and all that. I said, for one time, physics is on our side. Okay, so the last one is actually a surprising finding. We found later during the game that histotripsy also stimulates immune response. And I will talk about why, and that's the case later. But it's actually disrupt the cell membrane and then release tumor antigens. And anyway, so it has all this cascade of immune response stimulating. In selected patients, we have seen that, even tumors that's off target. So in this patient, those tumors are not targeted. Target tumor actually is not on this slice of imaging. But those non-target tumor, 8 weeks later, also had a substantial reduction. So in certain patients, the immune response is so potent that it actually shrinks, causing regression of the non-target tumors. So I made an effort trying to condense my 25-year journey in one slide. So bear with me. Okay. So 2001, I started as a PhD student and asked the question, can we actually find something like histotripsy? 2002, after a whole year of failure, I first observed histotripsy. And 2004, actually published the first paper on this. But I can tell you from my first discovery, the whole scientific community for almost 10 years saying this is impossible. So I will talk about that too. But we have to really invest heavily in terms of time and science to figure out the specialized device for histotripsy and also the underlying mechanism. December 2009, there is some event that happened. We co-founded HistoSonics. And in 2016, we did the first clinical trial of histotripsy. This is in prostate, benign prostatic hyperplasia, BPH, and it failed. Now, I don't actually think it was a failure, but we'll talk about that too. So HistoSonics at that time point almost closed. And we switched to a liver cancer. And in 2019, that was a big turning point. That was the first histotripsy trial in the liver and also the first trial in cancer. And the results were remarkable. They were just really good. And that allowed us to actually go to the multi-center clinical trial, Hope for Liver, which led to the 2023 FDA approval. And 2024, we started the clinical trial for renal tumor and pancreatic tumor, both going well and still ongoing. And more recently, already 3,000 patients treated. And Kelly talked about that acquisition. So I do want to know that Kelly said exit. Now I don't call it exit because we're still ongoing. The company is still doing our thing, and there will be a future real exit. But so 2001, I came in as a PhD student, and really trying to figure out what I want to do with my PhD. I remember clearly, Ahi Ludomirsky, he's a pediatric cardiologist. He came into our lab meeting and gave a talk, and he was talking about how he was treating pediatric patients with congenital heart disease, especially some babies. They're really sick patients, and his patients are dying on the surgical table because he had to perform very invasive surgery on these patients in their heart, trying to remove tissue. And he came to the lab, he said, can you invent a technology, non-invasive technology, that can allow me to perform a real surgery? He said, I don't need radiation. Thermal ablation like microwave or radiation or radiofrequency ablation doesn't work. I need something that can mechanically remove the tissue, but non-invasively, so that I can save my patients, those babies, those sick kids. It was so inspiring. So right after that, and I went to my PhD mentor, Charles Payne. I said, I want to take that song as my PhD project. This is just so inspiring. And Charles is like, are you serious? Like, you know what you're doing. And I was like, I think so, I have nothing to lose, right? So then that's my starting of my PhD career. And I can tell you, for a good portion of my career or for my PhD, definitely for the first year, I started my weekly trip to the local slaughterhouse, where in the morning, I call them when they slaughter pigs and cows, I go in and they allow me to go to their trash can, do dumpster diving for very fresh internal organs. And $5 I can fill in my trash can. Then I can come back to my lab and do experiments in water tank. So what you're looking at is a piece of pig heart tissue that I got from the slaughterhouse in the water tank. And this is after a whole year that failed, no success. I was getting some sort of desperate. And as engineer, when things doesn't work, the first thing I think is if you hammer something, it doesn't work, you use a bigger hammer. So I was like, okay, increase the power, right? So I was able to find this Asian amplifier, which was multiple power amplifier, and 1,000 watts connected to 380 volts power supply. So there's only one place on campus I can do that. And I claim my 4 feet of space with my water tank and a big tissue, heart tissue. And then the lab mates complained I was being too loud because I was using powerful ultrasound pulsing that they can hear. They're like, ultrasound, you're not supposed to hear it. Okay. So I turned to the pulsing frequency beyond our hearing range, which is 20 kilohertz. That's a human hearing range. By doing that, I have to reduce my pulse to very, very short, to microseconds. And typically you don't do that because just like ringing a bell, ultrasound is like it takes time to bring up to full amplitude. By making really, really short pulses, you're cutting off your power basically. So people don't do that for therapy. But I have unhappy lab mates, so I need to do something. So this is I want to show you this video from 20 years ago. So it's really kind of old. But what you will see is that trying to look at here, transducer is 10 centimeter away, and I'm aiming here. And you see smoke coming out? That's not smoke. It's actually micron tissue debris because it's in the water tank, right? It's in micron tissue debris and flowing out. And you are seeing what I saw 24 years ago. And I did the first time, I thought I was dreaming. Second time, okay. Third time, I said my rule of thumb is if it repeats itself 3 times, it must be real. So I took a video of it. And I got this beautiful hole. So I had to run to my mentor's office, Charles King's office, said, Charles, you have to come and take a look. I think something very special just happened. And this is, envision this. We use a device 10 centimeters away and basically generate a hole. And that is just non-invasive surgery, right? And I saw this before just to show that this is a micron debris. However, a month afterwards, I had to go to Charles' office again. And I said, Charles, I have good news and bad news. The good news is I was able to repeat the experiments again. It's real. The bad news is I think I just broke the most expensive device in the lab. And I don't think it's my fault. And I can't pay you because I was dirt poor. I was really, really poor. So I was like, I think the most powerful device, commercial device at the time, is not robust enough to do histotripsy. And that's what the community told me too. Like I presented to the scientific community, they said, you can't make it work. It's impossible because there's no device to basically produce the parameters we're talking about 200 times of the actual sphere connected in microseconds. So I told Charles, I said, Charles, I think this is something really, really special. If we are serious about it, we need to design and develop our own device. Right? But I was a PhD student, right? Like, what do I know? And he is a faculty. He needs to bet on his career on developing something that may not generate any outcome or grants funding for a long time. But he said yes. Charles always wanted to be a leader. He hates to be a follower, and he grabbed this. And I just want to show you that this was the amplifier I used in that experiment that I saw, as almost as tall as me, the transducer. And this is just 20 years later, our device showing this is one element, this is actually 260 element. of transducer built with 3D printing. And then we developed the amplifier, and this has 1 channel, and this has 8 channels, can fit on the size of 1 palm, and each one is 4 times higher power than this, and then can do not only therapy and imaging. Another thing that scientific community has been all telling me at the time that I was crazy, and this is not possible, is that the mechanism. So at the time I told them, I said, we think it's cavitation, we're generating bubbles, and which is called cavitation. And it was it was something that people know for 50 years, but I think it's a harmful effect because it's very stochastic and you can't control it. And I was telling them we can control it precisely, non-invasive surgery, and nobody believed. What we found out is that, we actually breathing air. So we all have nanometer gas pockets in our body. And we actually, if we apply, we think that really just 1 negative phase microsecond pulse and overcome the surface tension of the nanometer gas pockets, which is over 200 times an atmosphere. Then we can actually, in a controlled way, expand the bubble to the nanometer gas pockets to microbubbles. And what you are seeing here is actually the dark spots are microbubbles collapsed into one and filmed by a high-speed camera in this 3D gel embedded with the breast cancer cells, which are the white spots. So when the bubble actually expands, the breast cancer cells getting squeezed. When the bubble actually collapses, the breast cancer cell get pull apart. So really it's that local mechanical strain, if you can generate in a very controlled way with disrupted cells. And Charles King is here, and Adam and Eli, my former student, we work on this together. So, and I still remember to this day, I was, presenting to the community and basically, questioned, and people said it's impossible. And Charles said one thing, and that was the best advice I ever received. He said, well, everyone is making an effort to telling you it's impossible. And then you are on the verge of breathing. So just do the best science you can do. Everything else will follow. And that was great advice, and that's what I did. So 2009, something happened. I became a system professor, and we have this culture foundation. So what it is that culture foundation, they fund actually translational activity. So they will go to 10 big university, and they actually fund activity as you can go and get legal consultation on freedom of operation, right, and regulatory pathway, those kind of things. So they help fund part of the histotripsy activities. And they hired a consultant, Tom Davidson. And Tom is a very successful entrepreneur. So he invented and developed a harmonic scaffold. And it's a device that you can cut with ultrasound and coagulate at the same time. So 30 years ago, he actually developed this and was able to go through FDA approval and go through clinical use and everything, and sold to Johnson & Johnson 30 years ago for $750 million. So after that, he was going around and trying to look for good project. He was, an entrepreneur. So he came to University of Michigan through culture, and he talked to our group and saw histotripsy. He got so excited. He's like, this is harmonic scaffold of scaffold is 10 times. And he wanted to work with the team, and he wanted to go be a CEO, have a company on histotripsy and be the CEO of the company. So, you have Charles and me, we're academics. And we, at the time, had this kind of prototype in the lab that sort of treating the animals. No way, it's not a human. We had concept tested. We think it's a great idea, but the evaluation when you become a company is still pretty low because we don't have any human prototype. We don't have any really early clinical data or things that can help boost the value of this. So this is where I'm going to take my first poll and by the way, I don't know the right answer to the poll. I'm sharing this because I go through this journey. I still don't know if I made the right decision or not at any time point. So it's really, I think it's a personal decision. So the question I have here is, if you were at that situation, right? You're this new professor, right? and Tom Davidson came in and said that he wanted to start a company with you. Do you go and form the company with him, or you want to wait for the company to for your lab to have maybe a human prototype and collecting more data to increase the evaluation, so that when you form a company, you can have more stock shares. I mean, you guys don't need me to do on that. Anyway, actually, that's why I'm curious of hearing your role to see what do you do. So yes and no. On this, I'm just click so I can make it go away. So online they will collect, we can see the number, and here I just want to raise hands and make sure you're not seeing. So yes, raise your hands. Okay. No, raise your hands. Meaning you can still start a company, just wait later for evaluation. Okay, so I think based on my hands showing here is I see maybe 80% said yes. And online, how many? Percentage? Yes. What's the percentage or the 100%? 100% said yes. Wow. Oh, great. So I can tell you I said yes, because I feel that it was too good opportunity to miss. Although, and also because I was naive, I knew nothing about company, and Tom was so impressive. I was totally blown away by it. So here is, you can see Tom Davidson here, and then he hired Jim Bercholina as the CTO, and Chris Gibbons as the CFO, and Charles and me and Tim Hall, my fellow PhD students, we research partners here work together, and Will Roberts. This is related to prostate application, so he's the urologist. So, and Brian Folks, a physicist. So we together formed HistoSonics, and also Tom brought in VC that led by Jim Adox, who is, many of you know, the venture investor from here, and he raised $11 million as a Series A. So we have a company now. All right, so many things happened. Fast forward to 2016. We were looking at prostate as the first application. I know I started with sick babies and the heart problems. Then the VC said, older man with a prostate issue has a bigger market, and that's where you should go first. I'm sorry. I said the same thing. Apparently, well, we learn, right? Like when you start a company, you have to make money. So anyway, so the first application is benign prostatic hyperplasia, which is enlarged prostate compressing on the urethra, causing urination difficulty in, most of them when they are old enough. So we thought is that we can go in, use histotripsy to liquefy, right, the extra prostate tissue compressing on the urethra, and then urinate it out, right? It's very easy, and then you're done. And it just makes so much sense. And then we tested in animals in the canine model. It worked beautiful. So it really, we thought it was a great application. We have excellent animal results and started a clinical trial. And the phase 1 trial, we did it in 25 patients. And we were able to see cavitation microbubbles on all of them. So we know we were able to at least start histotripsy. And we see symptom relief. But it's transient. So, they feel better, but at a year, they can go back to the baseline. There were no signs of tissue liquefaction as we saw in our animal model. And, by the way, if you ask a scientist like me if this is a successful phase 1 trial, I would say yes. The safety is demonstrated. We didn't have any major complications. Patient even said it's not painful. It's more painful when they did the transrectal imaging. And we had some symptom relief, but it was a failure in the eyes of the VC. The reason is that we didn't achieve the liquefaction results as we hoped. We spent out $11 million at the time already, so there's no money in the bank. And the BPH market changed. When we started, there were that many competing technology, but in 2017, there are a lot more. So it's harder. The market is changing. It's harder. So at the time point, we will almost have to close up HistoSonics. So what's next? what happened is my actually lab was at the time was already studying liver cancer. So we have got really good results. So just show you a couple of things. And this is a movie. We're showing that in the pig, we can have histotripsy through the intact chest and generating cavitation, and just being able to treat even through the intact rib cage that any volume we want to treat. This is in the pig model. We also in different tumor models showing that this is actually in a rat liver tumor model. We had a tumor in the liver, and this is a particularly aggressive hepatocellular carcinoma. So within 2 to 3 weeks, the tumor will just grow all over in the liver. It's crazy, and we have to use nice rats. But in the treatment group, this is the same cohort, same batch. We treat 50 to 75% of the tumor, and within like a month, 9 of the 11 of them, we can't even find the tumor, not even the residual tumor we didn't treat. And the other 2 had a much slower growth. And the 3 months time point, we sacrificed the rats, and there's no tumor we can find in the rats. So it was, we had good animal results. And at the time, we also had Jim Adox, remember our chairman of the board, was engaging Johnson & Johnson for a long time. And able to finally got their interest, and they sent in a whole kind of group of people with their technical people, their clinicians, their management team, and finally able to convince them to come to our lab for a demo. And I remember that meeting because I was 9 months pregnant. The day before, I was sent to ER because I had a gallstone episode, and I kept telling them I'm not having a baby. And I knew I wasn't. I know that was my second. So anyway, so they were not doing anything to me anyway because it gave me painkiller. I was pregnant. So I told them, I said, send me home right away. I have something important to do next day. Anyway, so I was being a little crazy. But second day, I went to the lab, and I was able to give a really good presentation and a demo to the Johnson & Johnson group. And they really got impressed. And then, of course, they afterwards asked us to do some crazy experiments just to cover everything. And we were able to finish all that experiment, send to them, have further discussion. And when the BPH trial failed, they came back, said that they are willing and ready to put some money to HistoSonics, but for liver cancer, not for others. So we're like, okay, we're almost dying anyway. Let's switch to liver cancer. And then there's also a new management team. The new management team is actually so Tom and Chris Gibbon and Jim Bertrida at the time that they actually left, and the new management team came mostly from New Wave, which is microwave ablation company, and they have done liver cancer before, so they actually very knowledgeable. Also at the time, Charles Kane, my PhD mentor, got diagnosed with advanced prostate cancer. And I can tell you that before Charles and Tom have very strong personality. They really butt heads all the time. So there was always this issue as who takes the control in the company. So at that time point, a new management came in and said, they want to do this because they see promise, but they don't want the scientific team to be interfering with the company management running. And I, at the time, became the leader of the group, scientific group, because Charles was, sick, and we have to take care of him. So I was facing this, kind of challenge, and that was my goal too. Okay, I think he's still awake, so that's good. Goal two is that, as the leader of the scientific group, right, I was asked to give up control. And if we were you, would you continue seeking, would you give up the control? And knowing that the management team is good, like I didn't know them very well, but they have great track record. I talked to them, they seem to be good people, right? And you want to find a way to somehow have some, interference or influence so on. So the question here is, give up control, yes, and the other one will be. So people are doing this online, and I'm just going to click one so I can make it go away. That I'm going to ask you to respond. Okay. Yes, raise your hand. Okay, no, raise your hand. Oh, this time is more like, this time I would say 60-40. So 60 said yes and 40 said no. And it's similar online. Okay, so I can tell you I said yes to give up comfort. And the reason is that I feel that that kind of once I didn't have enough knowledge to feel like I can be a business person to really tell them what to do. And the second is that the early on that butting heads between the scientific team and the management team really was I feel it was really counterproductive. Because it creates this disfaction, and create like there's no trust. People just feel like we're fighting. And what I wanted is that I said I just want to stay connected so that I can check on the technical part. Like if you have any issue, you can come to me. I can answer all the technical questions. But I'm not going to have any interference or influence over your management business part. But the technology, is still my baby. I want to make sure technology is okay. And that part is not hard to do because I have a lot of formal students in HistoSonics doing the technology. But that's my personal choice. I choose to still working with the company, and because there's a licensing agreement and all that in place, but not really have any control over the company management side. So we switched to liver cancer, as I mentioned, that we had the first clinical system, and then we went to do this hopeful liver trial, and it's 2021, so COVID, okay? COVID happened, so it was not an easy trial to do, and also at the time, Charles passed away from prostate cancer. So we had this clinical trial in 44 patients in both the United States and Europe. It was not easy to recruit patients. And also, we had a majority of them with metastatic liver tumors, and a lot of them end stage. They tried everything else, everything failed, nothing works, and then they get enrolled in histotripsy. So this is just really brief data that we actually had 42 out of 44 had technical success, meaning that the clinician looked at the tumor, right? And they actually said, this is a tumor we want to target. And after treatment, they look at the imaging, and the imaging shows that the tumor with a margin that they defined actually did, work, and there's no, mistargeting and all that. So I was actually encouraged by that, because you have to understand, those are clinicians, knew nothing about history. They come in, they get trained, right? And then they said, go ahead and treat patients. Okay, so it's like a newly trained clinicians still able to get this kind of technical success, give me hope that the device and the technology is, relatively easy to use, and that there is a hope for wider adoption of this. And the safety looks great, and I'm not going to details. There are 3 adverse effects. Later turned out that they are not, they're actually caused by histotripsy. So the safety, they will say, below 7% of the adverse events. And this is just showing example of a patient where you can see like a year follow-up. So in year follow-up, we had still 90% of the regression control for the tumor that's treated, which looks good. And you can see that the debris absorbed by the body. So eventually you have some sort of maybe fibrous kind of nodule left there, but mostly it's absorbed by the body. So 2023 got FDA approval, and how much? 5 minutes. I just want to kind of talk about quickly the lessons learned. So what I learned from this journey about medical technology specifically, one is that the right regulatory path is very critical. So you have, 510K the lowest, then de novo, then PMA. We were able to do the breakthrough device track under the de novo and make so that we can have a smaller patient population than the PMA. So the bar is relatively, not as low as 510K, but lower. And being able to talk to FDA early on, especially for new technology, because you have to educate them. They don't know what it is. You think they are experts, but it's new, so they don't understand. So having that conversation to make sure they actually understand, so that they can, really design or adopt the right standards for you. That have been very helpful. And also being able to design the trial so the success bar is lower and not claiming something, like we go write publication. We like to claim this kind of wonderful things, but for clinical trials, not like that. You want to make sure you cover the safety, but don't claim too much for the efficacy. That will make your job and your life harder. So my last goal is on the cost we did. So how much do you think it costs for HistoSonics? And Kelly, I think you know the answer, so you don't get to go because that would be cheating. And I'm going to even click here right now because I don't want to mislead anybody. I'm just going to try to put it aside. How long do you think it costs for HistoSonics to get to where we started, which is really very minimal, to FDA approval and reimbursement, okay? So that you can do the first year sale. So we actually do the first year sale for liver tumor treatment. So A, B, C, and D. A, $5 million, B, $20 million, C, $100 million, and D, $200 million. So I'm going to just quickly go through. So A, okay, good. B, C, okay, D. All right. You guys are great. It's a room full of experts. The answer is D. And online here we got, I think, 65 or 70% of the answer D correct. So I was, you guys seem to be, okay with number. I was shocked. I was like, oh my God, if I knew it would take 20 years and $200 million to get to this, I didn't know if I would have started. But then I also, but I was sure that this is not uncommon for a brand new technology breakthrough, and this is actually about what it takes, okay? And then the second thing I learned is, when I was a PhD student, I thought, when did we get FDA approval? We're good, right? I achieved my goal. Now, after we got FDA approval, I was told, you're just starting. It's a new beginning. And then we have to do all these other things, reimbursement, super critical. And then I also told that before I was doing my PhD as scientist, we like to do this. We like to say in our presentation, in our paper said, we're going to change how medicine is practiced. Okay, we're going to change how, how this, the whole revolutionizing, right? And I turned out that clinicians hate that because they go to medical school, okay? Then they go to residency, fellowship, and they train for those so much, and so that they can learn how to take care of their patients with one way. And you go in and tell them, just forget about it. Now there's a new way because we're trying to change how medicine will be practiced. And you think they will just go along with it, right? And that's what I thought. But actually, they don't. So now we learned that if we want to reach a wide clinical adoption, you need to graduate doing it, starting where you need a clinical key opinion leader to champion you. Then also you need more and more people to come. It's a gradual process. And I'm not saying that we cannot get there. Honestly, if I knew any of this, I don't know if I would go to Charles King and say, I wanted that to be my dissertation. But you just don't know. So anyway, so lastly, last minute, I am setting up the world's first histotripsy center dedicated to research, education, and translation of histotripsy. So we're going to not only have workstations and personnel trained and have resources and funding to allow more researchers to do histotripsy here at Michigan. I already got space and everything. We also have an education program where we're gonna partner. So this is an informal partnership with HistoSonics. We also have partnerships so that we can incorporate histotripsy. Now it's going to be an official track in BME, so we can actually get students training this, and they're going to have internship opportunities at HistoSonics as part of the curriculum, so that later they will have jobs at least, right? So, then there's a translation part, which is tied with this innovation partnerships too. We're looking at new indications, so with different cancer applications, combine histotripsy with drug delivery, looking at neural applications, and also cardiovascular applications. So really a range, because it's a platform technology. And we do want to use this for many things. And also now with the histotripsy stimulating immune response, so we want to also combine histotripsy with immune drugs so that we can now work towards a systemic treatment as a curable cancer, not just as a local ablation treatment. And by the end, even though clinicians see it, I still want to see it again, that I think histotripsy is opening a new era of non-invasive surgery. And I believe that we will change how medicine will be practiced. Thank you. Yeah, I think there's a Q&A and I use something in common. So my strategy, I don't want to ask the question and I use polls. Ask my question, then I don't have to answer your question. So, Kelly, what do you think? Do you want to close it out? Okay, we'll take a couple of questions. Any questions? Yes.
I would love to know, you had the poll about giving up control in your company. What was your role before that? And then did you maintain any kind of role after that?
So the question is talking about giving up control of the company, and what's my role before that and what's my role after that. So I can tell you, it's here broadcasted. I'm still gonna say this. So before Tom Davidson was the CEO, right? And then actually Chris Gibbons went after Tom, and then Charles King was mainly representing the university side. So there was a lot of butting heads. So my role was everybody who came to me said, the other person should listen to me. Jing, you should tell them. So I was a mediator. At that time point where I was expected, or people expect me to try to bring the group together and have everybody listen to each other, and I was not successful. I eventually tell everybody they should just find their own therapist. And my role after that is that I was because of licensing agreement between university and HistoSonics. So first of all, I was leader of the scientific group, so our IT, right? All this I managed that. In terms of scientific findings, I was a scientific consultant with HistoSonics, so that I like when they have technical issue, they come to me and I consult. But what I learned was that you don't need a director role to have your influence. So you need to develop. The advice I got is that if you can have that trust and you can have this communication channel, then you can still exert your influence, right? So you don't need a formal role, but you can still exert your influence. So it's not saying that I gave up my gave up everything. I don't really interfere, but I was able to maintain a really good relationship with the management team. And on topics I feel it's important that I think I can contribute, I do reach out to express my opinion. Whether it's taken into account or not, I have no control over. But I do think now that relationship is still helpful. And now also I help promote, of course, histotripsy and HistoSonics product and many other things. But with histotripsy center, it's going to be more. Thank you. And thank you for all your support. Really, yeah.
Please join me in thanking our speaker.
Thank you. You have been great audience. Thank you.
Jen, thank you so much for kicking off this event for us. And we just have a small token of our gratitude. Thank you to everyone online for joining us as well. Have a great day.